For decades, scientists assumed that if you wanted to find the deepest, clearest genetic divisions between human populations, you'd look at the genes controlling the immune system. The genes responsible for how your white blood cells recognize and fight off disease. It seemed like the obvious place to look. Different continents faced different diseases for tens of thousands of years, so surely the genes built specifically to fight those diseases would have drifted apart the most, cleanly separating populations along the same lines people have used to define race for centuries. Scientists finally ran the actual comparison across every major population on Earth. What they found wasn't just unexpected, it quietly overturned one of the most common assumptions people have about how human genetic difference is actually organized. We're going to walk through exactly what that research revealed, step by step, and by the end, you'll understand exactly why a set of genes almost nobody outside of medicine has ever heard of turned out to hold one of the most surprising results in all of human population genetics. Stay with me, because this one genuinely reframes how deep racial genetic difference actually goes. If genetics, immunology, and the real evidence-based story of human biological diversity interest you, this channel is for you. Hit subscribe, because we dig into discoveries exactly like this one every week. Let's start with the genes, actually at the center of this story, because understanding what they do is essential to understanding why the result mattered so much. The genes in question are known as the HLA genes, short for human leukocyte antigen, a cluster of genes located on chromosome 6 that control how your white blood cells identify threats. Every cell in your body displays small fragments of protein on its surface, produced by these HLA genes, essentially acting like a molecular ID badge. Your immune system's white blood cells constantly scan those badges, and when they detect a fragment that looks foreign, a piece of a virus, a piece of a bacterium, a piece of a mutated cancer cell, they trigger an immune response to destroy the threat. There are actually two broad categories of these badges, and both matter for this story. Class 1. HLA molecules sit on the surface of nearly every cell in your body and display fragments from anything happening inside that specific cell, which is how your immune system catches cells that have been hijacked by a virus from the inside. Class 2. HLA molecules are found mainly on specialized immune cells and display fragments the body has captured from outside, allowing the immune system to mount a coordinated response against threats circulating through the bloodstream and tissues more broadly. Together, this system is essentially the body's entire early warning network for infectious disease, and its effectiveness depends almost entirely on how well it can recognize an enormous, constantly shifting range of possible threats. Because that badge display system has to be able to recognize an almost limitless range of possible invading pathogens, evolution has made the HLA genes some of the most variable, most rapidly diversifying genes anywhere in the entire human genome. While most human genes have relatively few common variants circulating in the population, some individual HLA genes have hundreds or even thousands of distinct known variants, an almost unbelievable level of diversity for a single gene, and by far the highest of any region in the human genome that's been mapped in this kind of detail. To put that in perspective, many genes elsewhere in the genome exist in just a handful of common variants across the entire human species. A single HLA gene alone can carry more documented variation than dozens of ordinary genes combined. That extreme diversity is exactly why researchers assumed, going in, that HLA genes would show the clearest, sharpest genetic differences between populations from different continents. The logic seems straightforward. Different regions of the world have faced different dominant diseases throughout history. Malaria in parts of Africa, particular viral pressures in parts of Asia, different pathogen environments in Europe and the Americas. And natural selection should, in theory, have pushed the HLA gene variants best suited to fighting off each region's specific disease pressures to become more common in that specific population over thousands of years, gradually creating populations with genuinely distinct, geographically clustered sets of immune-related genetic variants. To actually test that assumption, researchers needed to compare HLA gene variation across a genuinely global sample. Populations from sub-Saharan Africa, East Asia, Europe, the Americas, Oceania, and the Middle East, sampled in large enough numbers and sequenced in enough detail to actually map out how HLA variants were distributed geographically, and how much of the total human variation in these genes fell between those major continental groupings versus within any single one of them. This kind of large-scale comparative work built on decades of earlier, smaller HLA studies conducted originally for entirely practical medical reasons Organ and bone marrow transplant matching programs had been cataloging HLA variation within specific populations for medical purposes since long before anyone was using that data to ask broader evolutionary questions about how immune diversity is distributed across humanity as a whole. As those medical databases grew larger and more geographically comprehensive, and as genome sequencing technology advanced enough to characterize the full extent of HLA variation efficiently, rather than testing for only a handful of known variants at a time, researchers finally had both the data and the tools needed to run this kind of comprehensive worldwide comparison properly for the first time. Here's what the data actually showed, and it's the part that upended the original assumption entirely. The overwhelming majority of human genetic variation in the HLA genes, by most estimates, somewhere in the range of 85 to 90% of the total variation found anywhere on Earth, exists within any single population on any single continent, rather than between different continental populations. Put another way, two unrelated people sampled from the same country, the same city, even the same extended community, are typically more different from each other in their HLA genes than the average difference between two entire continental populations measured against each other. To make that concrete, if you lined up 100 people from a single city anywhere in the world and compared their HLA genes to each other, you would typically find nearly as much total genetic diversity in that one room as you would find comparing the average HLA profile of an entire continent against the average HLA profile of a completely different continent. The variation isn't concentrated between Africa and Asia, or between Europe and the Americas, the way it more noticeably is for certain other genetic traits. It's smeared almost evenly across humanity as a whole, present in roughly similar abundance inside essentially every community researchers have sampled. That result runs directly against the intuitive assumption most people carry about how racial or continental genetic difference works. It would be reasonable to assume that if you picked two people from, say, opposite sides of the world, their immune genes should look dramatically more different from each other than two people who grew up in the same neighborhood. The actual data doesn't support that assumption at all. For these specific genes, the genes most directly responsible for fighting disease, geography, and continental ancestry turn out to be a remarkably poor predictor of how similar or different any two individuals' immune genetics actually are. Why would this happen? The explanation researchers have converged on involves a phenomenon called balancing selection. And it's genuinely one of the more elegant concepts in evolutionary biology once you see how it works. Most genetic variation that gets shaped by natural selection follows a fairly intuitive pattern. A specific variant proves useful in a specific environment, so it becomes more common in the population living in that environment, while a different variant proves more useful somewhere else. And that pattern of local adaptation is exactly what produces most of the geographic genetic clustering researchers find when they study other, less immune-related regions of the genome. This is sometimes called directional selection, and it's the process most people picture when they think about evolution acting on a population over time, a population converging, generation after generation, toward whichever specific variant works best in its specific local environment. HLA genes work under a fundamentally different kind of selective pressure. Because the primary threats these genes are defending against, viruses, bacteria, and parasites, are themselves constantly evolving, constantly mutating, to slip past the immune system's existing defenses, there's a strong evolutionary advantage to maintaining as much diversity as possible within a single population, rather than converging on one single best variant. A population where everyone carries slightly different HLA variants is collectively far more resistant to being wiped out by any single pathogen. Because a pathogen that manages to evade one person's specific immune recognition system is far less likely to evade everyone else's slightly different version at the same time. This is sometimes explained using a concept researchers call negative frequency dependent selection. As any single HLA variant becomes more common within a population, pathogens face stronger and stronger evolutionary pressure to evolve specifically around that increasingly common variant, since doing so would let them infect an increasingly large share of the population. That pressure gives rarer variants within the same population a relative survival advantage, simply because pathogens haven't specifically adapted to evade them yet. The result is a kind of constantly shifting equilibrium that actively works to preserve rare variants rather than let them disappear, which is precisely the opposite of what happens to most rare genetic variants elsewhere in the genome, where genetic drift alone tends to gradually eliminate rare variants over enough generations, unless something is actively working to preserve them. Diversity itself, not any single optimal variant, is what actually gets selected for and preserved. which is exactly the opposite. Genes ended up looking so different from the rest of the genome once researchers actually mapped them out in detail. This is where the finding gets even more remarkable, because the same balancing selection pressure that maintains this extreme diversity within populations has also preserved something researchers call trans, species polymorphism, certain specific HLA gene variants that are so ancient and have been maintained under such consistent selective pressure for so long that some of them actually predate the split between the human lineage and our closest living relatives, chimpanzees, roughly 6 to 7 million years ago. Researchers first identified this pattern by building evolutionary family trees for individual HLA gene lineages, the same basic technique used to build a family tree for any gene, tracing how different variants of a gene relate to each other and roughly how long ago they diverged from a shared ancestral version. For the overwhelming majority of genes in the genome, that kind of family tree neatly separates into distinct human and chimpanzee branches, splitting apart cleanly around the same 6 to 7 million year mark when the two species themselves diverged, exactly as you'd expect. For certain HLA gene lineages, the clean split doesn't appear. Instead, some human HLA lineages sit more closely nested next to specific chimpanzee lineages than they sit next to other human HLA lineages, meaning the split between those specific variant lineages happened well before the human and chimpanzee species themselves ever separated. Think about what that actually means in practical terms. For certain specific HLA gene variants, comparing the genetic lineage of that variant reveals that some human beings carry a version more closely related, in evolutionary terms, to certain chimpanzee HLA variants than to a different HLA variant carried by another human being. That's not a typo, and it's not an exaggeration for effect. It's a genuinely documented finding in evolutionary immunogenetics, and it happens because balancing selection has kept certain ancient variant lineages actively circulating and useful for millions of years, long before modern humans existed at all, surviving essentially unchanged in their basic evolutionary lineage straight through the process of human and chimpanzee ancestors splitting into two separate species. The variant is older than humanity itself, and it's still doing its job today in real human immune systems, exactly the way it was doing its job in a shared human-chimpanzee ancestor millions of years before anyone alive today existed. It's worth emphasizing just how unusual this is across the rest of biology generally. Ordinarily, the moment two species diverge, their genomes begin accumulating independent mutations and drifting apart from each other, gene by gene, at a fairly predictable rate that geneticists use as something like a molecular clock to estimate when species split apart in the first place, for that clock to essentially stop, or run in reverse, for a specific handful of gene lineages, with certain variants staying recognizably similar across two separate species for millions of years after their common ancestor split apart, requires an unusually powerful and unusually consistent selective force actively working against the normal pattern of divergence the entire time. Balancing selection acting on disease resistance is, as far as researchers have been able to determine, essentially the only force strong enough and consistent enough over millions of years to produce a pattern this unusual. That's the result that genuinely surprised researchers studying human population diversity broadly, even though immunogeneticists studying HLA genes specifically had suspected something like this pattern existed for years before the full picture came together. It means that for this particular set of genes, arguably some of the most biologically important genes in your entire body, given how directly they determine your ability to survive infectious disease, the tidy, expected story of clean genetic separation between continental populations essentially breaks down. Race, as a genetic category applied to the rest of the genome, does correlate weakly with certain regional patterns in overall ancestry, but for the specific genes responsible for fighting disease, that correlation all but disappears, replaced instead by a pattern of deep, ancient shared diversity maintained across the entire species by a completely different evolutionary force. There's a practical, real-world consequence of this finding that makes it more than just a fascinating academic curiosity, and it's worth walking through because it's exactly the kind of detail that makes this discovery matter well beyond a genetics classroom. HLA matching is the critical factor determining whether an organ or bone marrow transplant is likely to succeed or be rejected by the recipient's immune system, because a poorly matched HLA profile causes the recipient's white blood cells to recognize the donated tissue as foreign and attack it, exactly the same way they would attack any other unfamiliar biological material entering the body. Given how the research on HLA diversity actually plays out, the best potential donor match for any given patient is not reliably predicted by shared race or ethnicity at all. A patient can, and often does, find a closer HLA match with a donor from a completely different continental background than with someone who shares their own broad ethnic category, precisely because of how thoroughly this specific set of genes scrambles the usual pattern of continental genetic clustering. Transplant physicians and matching registries have understood this in practice for years, even before the deeper evolutionary explanation was fully worked out. Successful matches routinely occur across racial and ethnic lines, while a shared ethnic background between donor and recipient is, on its own, no guarantee of an actual compatible match. This is part of why bone marrow and organ donor registries around the world specifically emphasize the need for the broadest, most diverse possible pool of registered donors, rather than assuming that recruiting more donors who share a patient's general ethnic background is the most efficient way to improve match rates. The actual genetics of HLA diversity mean that expanding the total diversity of the donor pool across every population improves match odds for everyone, in a way that simply recruiting more people who look similar to a given patient on the surface does not reliably achieve, because surface-level ethnic similarity is a weak predictor of the specific underlying HLA genetics that actually determine compatibility. That said, it's also true that certain specific HLA variant combinations do appear at somewhat different overall frequencies across different broad population groups, which is why registries still track and report broad ethnic representation among donors as one useful, if imperfect, piece of information. A patient from an underrepresented ethnic background in a given country's donor registry may genuinely face a harder search, on average, simply because fewer people carrying variants common in that broader population have registered as potential donors, even though the underlying biology means the eventual best match could, in principle, come from almost anywhere. It's worth being precise about what this finding does and doesn't mean, because a result this significant is also easy to overstate in either direction. This doesn't mean continental ancestry has no genetic basis at all. Plenty of other regions of the genome, unrelated to immune function, do show real, measurable, statistically clear patterns of geographic clustering that correspond reasonably well with continental population history, migration routes, and deep ancestry, patterns that consumer ancestry tests and academic population genetics studies alike rely on and can measure with real precision. Genes related to skin pigmentation, certain metabolic adaptations, and specific traits shaped by strong regional environmental pressures do show meaningfully different frequencies across continental populations, and that variation is real, well documented, and not in dispute. What this finding specifically demonstrates is that the genes controlling immune function, arguably among the most biologically consequential genes in the entire human genome for actual survival, follow a distinctly different evolutionary logic than most of the rest of the genome, one dominated by maintaining diversity within populations rather than by geographic divergence between them. It's a finding specific to this particular category of genes, not a claim that overturns everything researchers know about how continental ancestry is reflected in the genome more broadly. It's also worth being clear that this finding doesn't mean disease susceptibility is identical across every population either. Certain specific HLA variants and their frequencies genuinely do vary somewhat between different populations, and that variation has real documented relevance for public health, vaccine design, and disease research in specific contexts. Researchers studying malaria resistance, for example, have identified specific HLA and related immune gene variants that appear at meaningfully higher frequency in certain West African populations with long histories of exposure to malaria, reflecting real, measurable local adaptation layered on top of the broader pattern of shared global diversity. What the research shows is that this variation doesn't cleanly sort along continental or racial lines the way many other unrelated genetic traits do. The pattern is far more scrambled, far more locally variable within any given population, and far less predictable from someone's continental ancestry alone than most people would intuitively assume, precisely because balancing selection has spent millions of years actively working against the kind of clean geographic sorting that shapes so much of the rest of the human genome. There's a broader scientific lesson embedded in this particular result that extends well past immunology specifically. It's a powerful, concrete illustration of something population geneticists have emphasized for decades, but that remains genuinely counterintuitive for most people, and countering it for the first time. Human genetic diversity overall is overwhelmingly distributed within populations rather than between them, and different categories of genes can follow dramatically different evolutionary patterns, depending on what kind of selective pressure has shaped them over time. Skin pigmentation genes shaped heavily by regional differences in ultraviolet light exposure over tens of thousands of years, do show comparatively clean geographic clustering, which is part of why visible physical traits like skin color can feel, intuitively, like they should predict deeper genetic difference more broadly. If the trait you can see with your own eyes sorts so clearly by geography, it seems reasonable to assume everything else about a person's genetics should sort the same way. But visible traits like that turn out to be a poor guide to how the rest of the genome, and especially genes as biologically critical as the ones controlling immune function, are actually organized across human populations. Skin pigmentation involves a comparatively small handful of genes, shaped by one specific, geographically consistent selective pressure, sunlight exposure, acting in essentially the same direction for thousands of years in any given region. Immune function involves hundreds of interacting genes shaped by a constantly shifting, locally unpredictable landscape of pathogens under a selective pressure that specifically favors diversity over convergence. Those are two fundamentally different evolutionary stories playing out on two different parts of the genome at the same time, in the same populations, producing two completely different geographic patterns as a result. The genes you can see on the outside and the genes doing some of the most important work on the inside don't follow the same evolutionary map at all, a distinction that matters enormously for how confidently anyone should extrapolate from visible physical traits to assumptions about deeper genetic difference more generally. That mismatch is exactly why this particular finding carries weight well beyond a specialist immunology journal. It's one of the clearest, most rigorously documented pieces of evidence available anywhere in genetics for just how poorly the everyday concept of race, built almost entirely around visible physical traits, actually predicts the deeper genetic reality of how human biological diversity is organized, especially for the genes that matter most directly for something as consequential as your ability to survive disease. Two people who look nothing alike and who trace their ancestry to opposite sides of the planet can carry more similar, more closely related versions of these critical immune genes than two people who look remarkably alike and share the same regional ancestry. The genome simply doesn't organize itself along the same lines our eyes do. Researchers studying this pattern have also connected it to a much longer evolutionary story about how humans, as a species, have survived repeated waves of infectious disease across our entire evolutionary history. Every major population on Earth has faced serious infectious disease pressure at some point in its history, different specific pathogens in different regions, but the underlying evolutionary problem has always been essentially the same, staying one step ahead of rapidly mutating threats that are actively evolving to evade whatever immune defenses a population currently has. Historical epidemics offer a striking, if grim, illustration of just how consequential this diversity has been in practice. Populations with less diverse immune genetics, for whatever historical reason, have repeatedly proven more vulnerable to catastrophic population-wide losses when a new pathogen arrives that most individuals happen to share the same weakness against. A pattern researchers have documented in the study of several historical epidemic events where genetically more uniform populations experienced disproportionately severe mortality compared to more immunologically diverse populations exposed to the very same pathogen. Balancing selection acting on HLA genes appears to be one of the primary evolutionary tools that allowed the entire human species across every continent to keep meeting that challenge successfully enough to survive and expand as a species, generation after generation, epidemic after epidemic, for hundreds of thousands of years, which may be part of why this particular selective pressure has remained so consistently strong across every human population studied, regardless of which specific diseases that population's ancestors happen to face. Put the whole picture together, and what emerges is a genuinely striking correction to a reasonable but ultimately incorrect scientific assumption. Researchers expected the genes most directly shaped by the disease pressures different populations faced across history to show the clearest, sharpest genetic divisions between those populations. Instead, they found some of the most deeply shared, most anciently preserved genetic diversity found anywhere in the human genome. Diversity so old in some cases that specific gene lineages traced back before humans and chimpanzees were even separate species, maintained ever since by a form of natural selection that actively works to keep populations diverse rather than letting them drift apart. That's the real story behind a result that, on paper, sounds like a fairly dry technical finding about a specific class of genes, but underneath the technical detail sits a genuinely profound correction to how deep racial genetic division actually runs in the specific genes that matter most for something as fundamental as surviving disease. The category that shapes so much of how people intuitively think about human difference turns out to explain remarkably little about the genes doing the most important biological work of all. There's something worth saying within that. The genes controlling your ability to survive infection, the genes a doctor checks before an organ transplant, the genes evolution has protected more carefully than almost any others in your entire body. Those genes don't sort themselves by continent, by nationality, or by any of the categories people typically reach for when thinking about human difference. They sort themselves by an ancient, ongoing arms race against disease that every human population has been fighting together in parallel for longer than our species has technically existed in its current form. In the genes that matter most for staying alive, humanity has never really been as divided as the map might suggest. If this changed how you think about race, genetics, and how deep human biological difference actually goes, tell me in the comments what surprised you most. The 85 to 90% figure, the transplant matching implications, or the fact that some immune gene variants predate humanity itself. I read every comment, and it's usually where the next deep dive comes from. And if you want more of the real evidence-based story behind what your DNA actually reveals, subscribe now. We're just getting started.